Good afternoon. Welcome back to Virology. So today we're actually starting the reproduction cycle. We're going to take a virus into a cell, and over the next lectures, we're going to look at all the different things that happen in order to get new virus particles. And we're going to start today by talking about how viruses attach to cells and then get in. Now, you know already from our definition of viruses that viruses are obligate intracellular parasites. They need to get into a cell in order to multiply, but they're too big to just diffuse into the plasma membrane, right? A cell is circumscribed by a plasma membrane, and cells are too large, viruses are too large to get in. In fact, water molecules can't even get in. They have to be taken up by processes that we'll talk about in a bit. So virus particles are too large to diffuse, so they depend on transport pathways that exist in the cell in order for them to get inside. And so viruses bind to specific receptors on host cells, for the most part. There's some exceptions, which I'll tell you in a moment. But basically, we have lots of virus particles floating around, and they're just randomly colliding with cells. And there is a non-specific aspect of this interaction. There's random collisions and electrostatics, virus particles bumping into cells with no specificity, but now and then the virus will bump into the right receptor on the cell surface. Right meaning it can bind to it, and it will attach and initiate infection. So this is a really random process. That's why lots of virus particles need to be made, and not many of them are successful at getting into the cell. So this is all about finding the right receptor. It's really a random process. And the virus, there is no intention, right? The virus is just bumping around, and eventually it'll bump into the right receptor. And that just shows you how random it is. Now, receptors for viruses are essential for all viruses except those of fungi. You know what fungi are, right? The Last of Us. Actually, not a bad program. Fungi, there are viruses, but they never leave the cell. They stay, they reproduce in the cell, and then as the fungi divide, they divide with them. In fact, some fungal viruses don't even have capsids. They're just nucleic acid. Okay, so there are no receptors for viruses of fungi or plants. Plant viruses get into plant cells by some kind of mechanical damage. It could be an insect that is looking for sap, and it picks up viruses in the process or deposits some, or farmers running over plants with their tractors and contaminating them. So mechanical damage. No receptors for plant viruses and fungi, but all the other eukaryotes and prokaryotes on the planet, those viruses need to bind to cell receptors. And this is a relatively new field. As recently as 1985, we only knew of one virus receptor, and that is sialic acid. Sialic acid, you're going to hear that a lot. This is the receptor for influenza virus. Now, why did this change in 1985? Lots of enabling technologies, techniques that allow you to easily identify receptors, like recombinant DNA, the ability to clone genes, encoding specific proteins, monoclonal antibodies, that is, antibodies against one epitope, flow cytometry, the ability to take a monoclonal antibody and use it to purify cells that express that one epitope as part of a protein, of course, nucleotide sequencing, and then methods for reducing the amount of receptor in cells siRNA to knock down receptor RNA, and CRISPR-Cas, and related technologies to knock out the genes encoding receptors. So all these things we need to prove that a gene encodes a receptor. So for example, you purify a protein and you show that it binds the virus particle. That's a good first step. So here is the glycoprotein of HIV-1 in red there, binding its receptor CD4. So binding of a protein is good, but it doesn't tell you anything about infection, right? You need to have a biological process here. And so you should make an antibody against that receptor protein and show that the antibody will block infection. That's a functional assay. That's very good. Then if you can clone the receptor gene and put that into a cell that doesn't have a receptor and show that it confers susceptibility, that would be great. Remember what susceptibility is, right? The ability of a virus to bind to a cell. That's what susceptibility is. So if you have a gene for a receptor and you put it in cells and it confers susceptibility, that's really good. So here we have cells that don't have a receptor. They're not susceptible, and we've put in DNA from cells that have the receptor, and they're now making the receptor. So that's a great criteria. The only problem with that is sometimes you need two receptors, two different genes to get susceptibility. So putting one in isn't going to be enough. And that was a problem with HIV in the early years. It needed two receptors. And if you're just putting one gene in, it's not going to work. And finally, now let's say you have a gene which you think encodes the receptor. You've put it in cells, and it makes the cell susceptible. Then you could take another cell that naturally has that gene and disrupt it with CRISPR-Cas or some other technology and show that that blocks infection. It's a beautiful proof that a certain gene encodes a virus receptor. So these are some of the many things you can do to show that a specific gene encodes a virus receptor. So here's an experiment from the early days of SARS-CoV-2, which identified ACE2 as the receptor. Now remember, in the very beginning, we had patients with unusual pneumonia. We got bronchoalveolar lavage from them. We put them on cells. We showed the cells developed CPE. We showed the cells made proteins that are reactive with antibodies against coronaviruses. So it looked like it was a coronavirus. And many coronaviruses bind ACE2, angiotensin converting enzyme 2. So the same authors whose work we've been following did this experiment. They take HeLa cells, which do not produce ACE2, and they put in HeLa cells a plasmid encoding ACE2. So they do that. They use human ACE2. They use bat ACE2, that's the B, and they use mouse ACE2. And then they infect them with the virus that they've recovered from the patient. So on the bottom here, HeLa cells alone, we have the blue stain is DAPI, which stains the DNA, so you can see that there are cells there. And then we have in the second column, we have an antibody against ACE2, which is fluorescing green. And you can see the HeLa cells are negative. And then in the third column, we have an antibody to the viral nucleoprotein, which is fluorescing red. And you can see the HeLa cells are negative. So HeLa cells have no ACE2, and they are not infectable. Now we go back to the top, and we have HeLa cells that we've engineered to produce human ACE2. They make lots of ACE2 on the surface, green, and they can be infected with SARS-CoV-2. And if you merge, you can see the green cells and the red cells are the same cells, showing that ACE2 is the receptor. So this is a beautiful experiment because We know ACE2 is a receptor for some viruses. We have reagents. We have a plasmid encoding ACE2. We have antibodies and so forth. Second column is interesting. Bat ACE2, that also seems to serve as a receptor for SARS-CoV-2. Of course, depends on the species of the bat. There are many species of bats, and only certain species harbor these coronaviruses, so they pick the right one. But look at the third one, mouse ACE2. So we make HeLa cells with mouse ACE2. The protein is definitely expressed on the cell surface, the green, but when you infect with SARS-CoV-2, there's no infection, there's no red color. And you can verify that with a plaque assay if you'd like. So mouse ACE2 cannot bind SARS-CoV-2, which is an important finding because if you want to make a mouse model, this was relevant. You're not going to be able to do that. What you have to do, and what was subsequently found, is that you could make 1 or 2 amino acid changes in ACE2 of the mouse and get it to bind human SARS-CoV-2, or you could change the spike of SARS-CoV-2 and get it to bind mouse ACE2. Either way, we now have mouse models for SARS-CoV-2 for a long time. Now, receptors, of course, are on the plasma membrane for the most part. That's how they encounter virus particles. And they are, the plasma membrane, of course, is a busy place, has lots of transmembrane proteins of different sorts, has integral membrane proteins, lipid-anchored proteins. It has carbohydrates attached to these proteins, of course. And then below there is the cytoplasm. And many of these have cytoplasmic extensions. Here on the bottom is just an example of some of the cell proteins that we know bind specific viruses. And that includes, here are the virus names at the top here. All different kinds of proteins. We have actually heparan sulfate proteoglycan. So this is basically an amino acid chain with extensive glycosylation. Sialic acid can be receptors for viruses, including influenza virus. Interestingly, SV40 binds to a glycolipid. So sugar is attached to a lipid molecule. The virus attaches to the sugars. Then we have a row of protein receptors, including the poliovirus receptor, CD155, which was discovered in my lab in 1990 here at Columbia. Herpes virus, foot and mouth disease. Here are the 2 receptors for HIV-1 and the 2 receptors for adenovirus. So those 2 viruses require 2 different cell proteins to get into cells. And remember, none of these proteins exist for the virus, right? They exist in the cell, they have cell functions, but none of them exist to take up virus and have the virus kill cells. That doesn't make any evolutionary sense, right? Even if the virus didn't kill cells, it still wouldn't make much sense. But these are all there for cell functions, and they happen to be, over time, co-opted as virus receptors. And these are just a few of the many, many, many. Now, different viruses can bind the same receptor. This is kind of interesting. So adenovirus here, which we've encountered already, and Coxsackie virus B3 bind the same receptor. And the swine herpes virus called pseudorabies virus here binds the same receptor as human poliovirus. So many years ago, Coxsackie virus, by the way, was discovered in Coxsackie, New York. Exit 21B on the New York State Thruway. Many years ago, I was driving up there and I took a picture because I had to show it in my classes. I actually have a listener who's from Coxsackie. She's very proud to be. In the old days, they would name viruses after where they were isolated. Now that's a no-no. People don't like that anymore. And we'll encounter it a few times. But back then, in 1947, like many things in the world, you just did what you were told. And that was it, Coxsackie viruses. Now, sometimes viruses of the same family bind different receptors. So rhinoviruses bind 3 different receptors. We'll look at that later. Retroviruses, 16 different receptors. And look at this, herpes simplex 1 has a lot of glycoproteins on its surface, and it can bind a number of cell surface proteins. And you may say, why do you need to bind so many receptors? Well, it could be that in a certain tissue, one receptor is important, and in another tissue, another receptor is important. Okay, so there's no rule. There's not one virus, one receptor, obviously. So let's talk briefly about how virus particles attach to receptors. And we have two picornaviruses here, poliovirus and rhinovirus. And these illustrate two different ways. So poliovirus, these are both built with icosahedral symmetry. So you can just see the five-fold axis of symmetry for poliovirus and for rhinovirus there. And for poliovirus, there is a groove around the five-fold axis. So here on the lower left is kind of a cartoon of the virus particle. And here in blue is the five-fold axis. Around it is a little groove. And in fact, the receptor, the N-terminal domain of the receptor fits into that groove. That's how the receptor attaches to that particle. But not all receptor-virus interactions have to have this lock and key aspect. There's rhinovirus on the right, which also has this groove, but the receptor doesn't bind to it. The receptor, which is shown in gray, binds at the five-fold axis. So if you look at this five-fold axis on this diagram, you can see there's kind of a flat region or plateau, and that's the region to which the rhinovirus 2 receptor binds. So that's in gray. And you can see how many receptors are bound. There are 12 five-fold axis of symmetry. Yeah, the gray part here, these are the receptors for poliovirus. So this is the structure of the receptor in a soluble form attached to the virus particle. And so you can see the difference in the topology of the receptor. This is a very rod-like protein, whereas the receptor for rhino are just sitting flat on the virus particle. Yeah. This structure was done in my laboratory many years ago. It's on my phone because I think it's so cool. It's been in multiple iterations of phone. I have many polioviruses. I have the lock screen here has polio. That's bound to an antibody. And then we have another one where the virus is broken open. No pets, no kids. I have viruses on my phone. Okay, now, that was an icosahedrally built particle. But another one, adenovirus, does it very different. So adenovirus, icosahedral particle, bigger, remember, bigger T number. It has these fibers sticking out from each five-fold axis of symmetry. And those fibers bind to the receptor. It's one of these specialized proteins. So in A is a structure of the fiber itself. So there's a base that's stuck into the particle itself, and then the shaft, and then a knob. And the knob sits down on the receptor. So the knob here is green, red, blue. And I see red and blue here. I don't see green, but basically CAR is the receptor in magenta here, here. And the knob is sitting down right on top of it. That's how it attaches, as you can see there. And so this is one of these viruses where two receptors are needed. First, there's a fiber receptor, which is CAR. And again, CAR is Coxsackie adenovirus receptor. They share the receptor. And then there's a second receptor needed for internalization, as we'll see later. So in other words, the receptor doesn't have to sit right on the surface of the particle. There can be extensions like this, and those attach to the receptors. So those are viruses with icosahedral protein shells. Now we're going to talk about viruses with membranes. It's a different story because whenever a virus has a membrane, it has glycoproteins stuck in the membrane. Otherwise, it wouldn't be able to do anything. So here's influenza virus, and the membrane is kind of pink. The RNA, the nucleocapsid is inside, and there's the membrane in pink. The line doesn't really point to the right thing. It should be pointing to that pink circle. That's the membrane. And the membrane is embedded with glycoproteins, including the hemagglutinin and the neuraminidase that we'll talk a lot about in this course. The hemagglutinin is the protein that attaches to the cell receptor, HA. And here's the structure of HA right here. It's actually a trimer on the particle, but I'm showing you a monomer for simplicity. There's the membrane of the virus, and we have an extended alpha helix, which makes the stem. And then we have a globular head. And if you look down onto the globular head, there's a pocket, and that's where sialic acid binds. So sialic acid is the receptor. I'm going to show it to you in a minute, but it's here in green, and it's interacting with various amino acids in that pocket. So that's how the HA of flu attaches to sialic acid via this pocket. Okay, so what is sialic acid? It's a sugar. Proteins can have sugars attached to them. They're called glycoproteins, right? And these little balls are different sugars. And the terminal one with the red arrows, that's sialic acid. When sialic acid is present, and it's present on a lot of glycoproteins, it's always the last sugar on a chain of sugars. And that's what influenza virus binds to. And there are two different flavors of sialic acid, and different kinds of influenza viruses bind to them. And so here on the bottom, so sialic acid in both is the yellow molecule. So there's your 6-carbon ring. And so this is sialic acid. And then this is the next sugar in the chain. It happens to be galactose. And in this case, sialic acid is attached to galactose by an alpha-2,6 linkage, which simply means the alpha, the number 2 carbon here in sialic acid is attached to the number 6 carbon in galactose via an oxygen. So that's an alpha-2,6 linkage. That's the kind of sialic acid that human influenza viruses bind to. Okay? That's why we have 2 humans here. This is the kind of linkage. Now, avian influenza viruses, including H5N1, which is all in the news, they bind alpha-2,3 linked sialic acids. So it's simply alpha, the number 2 carbon on sialic acid linked to the 3 carbon instead of the 6 by an oxygen. Alpha-2,3, human viruses, human influenza viruses do not bind alpha-2,3. They bind alpha-2,6, but avian influenza viruses bind alpha-2,3. And what people are worried about with H5N1 is whether the virus will change to recognize alpha-2,6 sialic acids. That would be the first step on getting a really good human infection. As you know, in the US, H5N1 has infected cows. You've heard that, right? A lot of dairy cows. Cows are fine. People who work with cows and get infected are fine. Those viruses don't bind alpha-2,6. They bind alpha-2,3. It turns out you have alpha-2,3 in your eyes. And so these people get conjunctivitis from the influenza virus, a very mild infection. And it's rare that it gets down into the lung. But we'll talk about that later. But so this is a good example of how receptor interaction can control the host range. What animals are infected by the virus? Here's an example of HIV attachment. There's HIV particle. It's enveloped. They're the glycoproteins. There are very few of them. It's sparse compared to influenza virus, where there are a lot of them, right? And this envelope, this spike is called envelope protein, and one of its receptors is CD4. And so you can see CD4 here in purple, and the viral glycoprotein binding to it here. Actually, there's a pocket in the glycoprotein of the virus right up here. It's expanded down here. And there's a tyrosine in CD4. You can just see there, the tyrosine. That's essential for binding. If you change that tyrosine, it won't bind anymore. It's quite remarkable. So just different ideas of how viruses attach to cell receptors. Here's how a spike of SARS-CoV-2 attaches to its receptor. We already said The receptor for SARS-CoV-2 is ACE2. Right, so here are some other coronaviruses. You can see NL63 binds ACE2. So does SARS, the first SARS. And there are 5 common cold coronaviruses that cause common colds, upper tract infections, and that's ACE1 is the receptor for one of them. So anyway, this is the spike protein for SARS-CoV-2. So it's embedded in the membrane, of course. And this is a linear diagram of the spike protein. 1,273 amino acids. That was the name of the vaccine, mRNA-1273. That's the reason it got that name, because it's the number of amino acids in the protein. Both Moderna and Pfizer, 1,273 amino acids. And this is the structure of the spike protein here on the right. And the receptor binding domain is in green, and all that is that's the part that binds ACE2. And ACE2 is shown in the right-hand structure here in red. So that's the interface between ACE2 and SARS-CoV-2 spike. We'll talk a little bit about this in a bit in some more detail. First question is, virus receptors on the cell surface can bind directly to icosahedral virus capsid proteins, interact with glycoproteins of enveloped viruses, can be carbohydrate or protein molecules, have cellular functions, all of the above. Okay, what did we do? Yeah, all of the above, right? So receptors can bind to icosahedral capsid proteins. They can interact with glycoproteins. They can be carbohydrates. They can be protein, like sialic acid, right? It's a carbohydrate. And they obviously have cellular functions, so all of the above. So now let's talk about how these viruses get into cells. We've talked about how they bind very briefly, icosahedral and envelope viruses, how do they get into cells? So cells has lots of mechanisms for getting things into them. And they're illustrated here. We have phagocytosis, where you can take up large particles. Macrophages and dendritic cells can do things like that in other cells. And then we have endocytosis in the middle. And there are a couple of forms of that. There's macropinocytosis. So let's assume this is water, these little dots. They're just being taken up into membrane invaginations, which pinch off into vesicles and go in the cell. So as I said, water cannot diffuse in, so it has to be taken up by specific ways like this. And this is not a very specific process. The water molecules are there, they're just grabbed by these membrane ruffles. And then we have receptor-mediated endocytosis, where a ligand will bind a receptor, and then the ligand-receptor complex is taken up into the cell by endocytosis in vesicles. This is part of a normal pathway to internalize molecules that are needed, and then they can be released. Then finally, there's a process called surfing, where cells have these filopodia, these extensions that contain actin, and virus particles can bind them and move down the actin filaments into the cell. So one of the key things you have to remember is that things don't diffuse in cells. So here we have this picture of this virus just kind of diffusing here. Nothing happened if the cell is too crowded. So here's a nice diagram by David Goodsell, the artist. And we're going from the plasma membrane to the nucleus in 4 panels. So we have our plasma membrane, and we have our actin microfilaments. We have microtubules. I see some ribosomes here. And then there's some endoplasmic reticulum. There's Golgi. And then the third panel, here's a nuclear pore. So something's going in the nuclear pore. And then, of course, in the nucleus, our DNA wrapped around histones in the form of nucleosomes. So the cytoplasm is really crowded. Things do not diffuse. People have calculated that if a virus particle bound to the plasma membrane, it would take hours to diffuse to the nucleus. if it were just random diffusion. So that doesn't happen because the cytoplasm is crowded. So how do things move? By motors. There are motors in the cytoplasm. And we're going to show you how that works. But here's an overview of the different ways that virus particles can get into the cell. So on the left, we have entry at the plasma membrane. Some envelope viruses, like this is a herpes virus. The membrane can fuse at the plasma membrane, then the nucleocapsid can move down the microtubule by motors until it gets to the nucleus, which is where it needs to go. We also have an adenovirus entering at the plasma membrane, the capsid being transported initially in an endosome, and then coming out of the endosome being brought by motors, dynein motors, down to near the nucleus. And other kinds of endocytosis, clathrin-dependent endocytosis, where particles are brought in these vesicles and then liberated. But in all cases, the vesicles themselves or the particles are always carried on these microtubules using motor proteins that require energy. So here's another kind of endocytosis. The vesicle is moving on the microtubule. So the point is there's no diffusion. There's always motor-driven transport. So I think we have an animation of one of these. This is an endosome being brought into the cell on a microtubule. That's that long protein. And that's a motor protein walking down the microtubule, and this requires ATP. So that if that has a virus particle in it, it's bringing the virus particle into the cell. So there's a company called Ex Vivo. They do very nice animations of this sort.
It has a good walk.
Sorry?
It has a good walk.
It has a good walk, yeah. It's got a little funky to it, yeah, I agree. Okay, so let's talk about viruses that enter at the plasma membrane first, and then we'll talk about those that get in via endocytosis. So here's an example at the top of a virus that is binding to a receptor on the plasma membrane, but then it fuses immediately. The membrane of the virus fuses with the plasma membrane, so now we have the viral nucleic acid in the cytoplasm. So how can this happen? This is a highly regulated process because, virus particles bump into things all the time, and you don't want them fusing with anything. They need to fuse with the right cell. So how do you do that? Well, it usually is via a receptor. And there are two mechanisms I'm showing you here for that. So first, here's a paramyxovirus, which would be like measles virus. This virus has an attachment protein and a fusion protein next to it. And when the attachment protein binds the cell receptor, which is in purple, it undergoes a conformational change, which then causes the fusion protein to flip out and stick into the membrane. So fusion proteins are typically like 20 amino acid long, highly hydrophobic sequences, which can stick in membranes. Membranes are very greasy, right? And you need greasy amino acids to stick into them. And the key to fusion of two membranes is to get them really close and get rid of the water molecules that are between them. And when you have a fusion protein like that sticking in, it can do that. Now the key here is that the fusion protein is hidden until the receptor, the virus attachment protein attaches to a receptor. Then that causes a conformational change to expose the fusion protein. So in other words, the virus will not fuse with a cell that doesn't have the right receptor. So it's regulated. So that's one example using one cell receptor. Then on the bottom we have HIV-1, where two receptors regulate this activity. So here we have the virus with its glycoprotein GP120 and the fusion peptide. Again, it's hidden, it's bent up towards the virus, so it's not going to be fusing with anything. This GP120 binds first CD4. That causes a conformational change in the viral glycoprotein, which now enables it to engage a second receptor, like a chemokine receptor here in purple. And then when that's done, the fusion protein flips around and sticks into the cell membrane, and that will bring it close together so that it can fuse. So the key here is that fusion's regulated. It's regulated in this case by either one or two receptor interactions, because you don't want this fusion protein just flipping around, fusing with anything it bumps into. That would be very inefficient, right? You want it to fuse with cells that have the right receptor. All right, here's an animation of HIV-mediated fusion. It always goes too fast. So here is the viral glycoprotein, right? And here it is in a trimer form. It's stuck in the cell membrane. So the viral membrane is down here, and the cell membrane is up here. So now the fusion peptides have been extended due to the second receptor interaction. These proteins don't like to be in this conformation. It's not energetically favorable. So they're reorienting, and in so doing, they're pulling the membranes close together, and eventually they're going to fuse, okay? And first the outer leaflets will fuse, and then the inner leaflets will fuse, and you will have a pore going through them. But this, let's see if I could back up just a bit. Yeah, that's the second pore forming, and now the contents of the virus could go through into the cell, complete fusion. So why do the glycoproteins do that? Let me try and show you. So this is called the extended conformation, which is what they do when the second receptor interaction occurs and the fusion peptide extends up into the cell membrane. The protein does not like to be in that conformation. It's energetically unfavorable. And so it returns to what it likes, and that involves a process we call hairpinning. But essentially, if you notice, the alpha helices are forming along the other alpha helices, and that makes the two membranes come together. Okay, so that's called hairpinning for reasons that, well, it kind of looks like a hairpin. You'll see that better with influenza virus. Okay, next question. Which of the following does not play a role in virus entry? Clathrin-mediated endocytosis, fusion of viral and plasma membranes, the fusion of virus particles in the cytoplasm, microtubule-mediated transport, sialic acids, which does not play a role in virus entry. Okay, let's see how we did here. Yes, diffusion doesn't play a role. Almost everyone got that. Fusion of viral and plasma membranes for sure plays a role, right? Endocytosis plays a role. Microtubule-mediated transport, sialic acids can be a receptor. So the one that doesn't is diffusion. Okay, now influenza virus will make this hairpin idea more concrete. So the influenza virus is shown here binding to sialic acid receptors at the top, and it's being taken up into an endosome. And eventually the viral RNAs are going to come out. So we're going to follow that process. Now here are two hemagglutinin molecules close up. The gray heads, those are the globular heads. Those are binding the sialic acid on the cell surface. And these HA molecules are trimers. Most of these fusion proteins are trimers. Now, if you look at this HA, I don't know if you can see, but there are some red sequences down by the virus membrane, there, and there, and there, and there. Those are the fusion peptides. They're hidden by the virus membrane, so they don't fuse with anything. As the particle moves into the endosome, the pH drops. It's a normal process. Protons are pumped into the endosome. As the pH drops, the HA reorients and the fusion peptides get thrust up into the cell membrane. So they go from near the virus membrane, they go up into the cell membrane, very much like HIV we just talked about. This, you can view this intermediate. You can see now the fusion peptides in red as taking a hairpin and extending it, right? So here's a hairpin on the right. So the influenza HA is a hairpin. It's folded back on itself. When you drop the pH, you extend this hairpin, you basically pull it apart there. And it's not very happy as a extended hairpin. So it starts to hairpin to go back to the hairpin configuration, and that's what's shown here. So the the alpha helices begin to reform. They've been extended. They begin to reform here. That pulls the membranes together, and they fuse. But now they're in the native conformation, the hairpin conformation. They've reached their happy energy level. And in so doing, they brought the two membranes really close together. So again, they're in a hairpin conformation. Low pH extends it to push the fusion peptides into the host cell membrane. And then they slowly re-hairpin because it's not favorable to be extended. That brings the membranes together, and then they fuse. Same thing happens with HIV, but here it's much clearer. And when the membranes fuse, the virus and the endosome membranes, the RNAs come out, the nucleocapsids of the virus come out, and they go into the nucleus in order to replicate. I think we have an animation. Yeah, we do, but it's after this. So these are the three states of the HA. So there on the right is the post-fusion state. That's the extended state with the fusion peptide in the cell membrane and this part in the virus. This is the unfavorable part. The middle one is the favorable part, the way where the two alpha helices you see are folded over each other like a hairpin. The fusion peptide is hidden down here. So acid pH is needed to extend this and thrust it into the cell membrane. There's one other regulation besides acid pH, and that is protease cleavage. The fusion peptide is not free at the N-terminus until a protease comes along and cuts it. So if you subjected this molecule to low pH without cutting it, the fusion peptide would not be able to reorient. So these peptides, these proteases cut the fusion peptide. Now you have an N-terminus that is hydrophobic, and when that gets thrust into the cell membrane, it can insert into it. So low pH and a protease are needed to control the fusion of influenza virus. Because again, you don't want it to happen anywhere. All right, here's the animation. That's influenza virus attaching to sialic acids. It's being taken up into an endosome. And these are clathrin-coated endosomes. Those are specific proteins on the outside. And it will move on a microtubule. There you go. Little motor. This one has a different, this is very frenetic, this motor. It's really in a hurry. The pH is dropping and the membranes are fused. You don't see any of the details, but the membranes are fused. Those are the ribonucleocapsids of the virus that come out and they enter the nucleus. Okay. These fusion proteins I've been telling you about, they're called class 1 fusion proteins. They're perpendicular to the membrane. They're mostly alpha helical, and they're trimers, and they include flu. We've talked about HIV and some other viruses, simian virus 5, Ebola virus, and Maloney murine leukemia virus. These are all type 1 or class 1 fusion proteins. Now, I wanted to tell you a little bit about SARS-CoV-2 entry to prove to you why hydroxychloroquine never would have worked, no matter what. You know the story with hydroxychloroquine, right? So a group in France did some experiments in cell culture and found that hydroxychloroquine would inhibit reproduction of the virus. So without any clinical trials, the president at the time forced the FDA to license this drug to treat people. And it didn't work, and it killed a lot of people because it has bad side effects. The reason you could do that is because it was licensed as an antimalarial, so you could do an off-label thing. But it didn't work. To this day, you will find people who swear that hydroxychloroquine works, and Big Pharma is just keeping it from them because they want to make a lot of money. My God, it's so dumb. Look, here's why. Just look at the biology. In the cells and culture that those people used, the virus binds to ACE2, gets taken up by endocytosis, and there's a protease in the endosome called cathepsin L that cleaves the spike and catalyzes fusion within the endosome. Hydroxychloroquine inhibits cathepsin L. That's why it inhibited reproduction in cells in culture. Your lung cells, this is not how it gets into your lung cells. In your lung cells, it gets in at the cell surface because there's a protease called TMPRSS2 that cleaves the spike and allows fusion at the plasma membrane. So hydroxychloroquine has no chance to get near the virus particle. And that's why it would never have worked. All you had to do is read a paper or two, or take this course, listen to this lecture, because the first year I talked about this, but nobody listened. And to this day, they're not listening. And this is really sad because fewer and fewer people are listening to science. But that's why hydroxychloroquine would never work, because its mode of action is in the endosome, not at the plasma membrane. This is the way the virus gets into your lung cells, not by endocytosis. But in any case, what's interesting is that the cleavage of the spike allows the fusion peptide to be flipped up, very much like low pH would, but we don't need low pH here. We just need cleavage, and boom, here's the fusion peptide here, FP. It's right there. It's buried in the native protein. Upon cleavage, there it is at the tip of the protein, and it can now cause fusion at the plasma membrane. So similar principle, but it's cleavage that is causing the conformational change, not low pH. It's why hydroxychloroquine never had a chance. There are also class 2 fusion proteins that are flat on the surface of the virus particle. They're mostly beta sheets compared to class 1, which are alpha helices. They're dimers instead of trimers. They're parallel to the membrane. But they still work. And they still work like hairpins. So here we have a flavivirus with these fusion proteins laying flat on the virus surface. Here it is in a cartoon form in B. And again, the fusion loops are buried here in this protein. And when it undergoes low pH in the endosome, as the virus is taken into the endosome, these proteins reorient. The fusion loops go into the cellular membrane. They're unhappy in this extended conformation, so they hairpin, which means they reform their structures that are favorable. And in doing so, the membranes are brought very close together and they fuse. So that's the hairpin conformation that is energetically favorable. So the low pH drives it out of the hairpin conformation, but it can't keep it there, and it slowly reforms, and that's what drives fusion. I think we also have an animation here. This is dengue virus entry coming into an endosome along a microtubule. pH is dropping. If you look inside now, that's the virus particle. As the pH drops, those glycoproteins are going from flat to extended, and the fusion proteins are at the tips of those extended molecules. They insert into the cell membrane. Those are the fusion peptides up there. And what's going to happen they're going to reconform to the favorable conformation, and in so doing, bring the two molecules together. And then they fuse, and then the viral RNA can go through out into the cytoplasm. I don't think the artist quite got the hairpinning right there, right? They need to talk to me. But it just isn't really hairpinning, but you get the idea. Okay. One more interesting twist on entry, and that's Ebola virus, these long filamentous particles that they don't seem to be binding any specific plasma membrane receptor. They seem to be taken up by that micropinocytosis, that cellular drinking, which randomly brings things into cells. So they're brought into an endosome, and in the endosome there are again enzymes like the ones involved in hydroxychloroquine that cut the glycoprotein, and they expose a binding site for a receptor in the endosome. So this blue protein here labeled NPC1, that is the endosomal receptor for Ebola viruses. When the virus binds those, the membrane fuses and the RNA comes out, as you can see there. We're not sure the mechanism of fusion, whether it involves hairpinning and so forth. It hasn't been studied that well. But what's unique here is that there's an endosomal fusion receptor. And that particular protein, by the way, NPC1 stands for Niemann-Pick cholesterol transporter. So this is a protein involved in cholesterol transport. And in fact, a number of people have genetic defects in the gene, and it's a very serious disease, and they end up not living into their teens. But cells from them were originally shown to be resistant to Ebola virus infection, proving that this was an important receptor for virus entry. Okay, so remember, fusion is regulated. If you remember anything from today, fusion is regulated because it can't happen in the wrong location. It can occur at neutral pH at the plasma membrane, where you need a second protein to regulate fusion. Remember, a receptor protein will help change the conformation to allow the fusion protein to be exposed. So at neutral pH at the plasma membrane, the second protein interaction is regulating fusion. We also have low pH mediated fusion, where we have virus taken up into an endosome. The pH drops, and that changes the conformation of the glycoprotein exposure to the fusion receptor. There has to be cleavage of the glycoprotein in order for that fusion peptide to be exposed. And I told you about that for influenza virus, for example. For SARS-CoV-2, the cleavage can happen at the cell surface. And then finally, another mechanism of regulation would be to have an endosomal fusion receptor. So the Ebola viruses are not going to be fusing all over the place, because that endosomal fusion receptor is only in the endosome. So that's the only place they will fuse, and to get there, they have to be taken up by endocytosis. Next question is, viral fusion peptides are exposed for insertion into the host cell membrane when the virus particle is near a cell, the virus particle is in the cytoplasm, trimers of the fusion peptides form, the endosome becomes acidified, the virus is docked onto the nuclear pore. When are the fusion peptides exposed? This is a tricky question. Be careful. Hey, how did we do? Well, the right answer is the endosome becomes acidified. That's what flips up the fusion peptides, right? The virus particle is near a cell? Really? It's just going to be near any cell and fuse? No, that's what I've been telling you. No, it's not when it's near a cell. It's in the cytoplasm, kind of late by then, right? No. Trimers form. Trimer doesn't really matter in the scheme of regulation. It's when the endosome becomes acidified. Dock on the nuclear pore, that's way downstream. That's not going to be it. So when the endosome, remember, the acidification causes the HA to flip up so the fusion peptides can go into the cell membrane, and then they hairpin to come back together. So it's the acidification that's the key. Okay, a couple of more things. How do you get the genome out of a non-envelope virus? We've talked about envelope viruses. It's easy. The membranes fuse, the genome comes out. This is adenovirus. Adenovirus binds to receptors. It's taken up by endocytosis. As the pH of the endosome drops, there are these little proteins. They're yellow triangles or diamonds. They come out of the virus particle and they poke holes in the endosome membrane. And those holes, then the virus particle comes out of those holes and goes down microtubules and docks onto the nuclear pore. And then the DNA comes out. So this low pH removes this, I guess, membrane solubilizing peptide. And here's some EMs. Here's an adenovirus on a microtubule, which would be this part. And here, adenovirus is docked onto nuclear pores. Now, an interesting thing happens at the nuclear pore. So remember, the adenovirus is motored down the microtubule by dynein motors. And it's already partially disassembled, right? And then the adenovirus binds to the nuclear pore, which is right here. But the kinesin motors are still stuck on it, and they start to go the other way, and they basically break up the capsid because the capsid's stuck on the nuclear pore and the motor's pulling the other way, and that breaks up the capsid. So now the DNA, which is shown here, can get into the nucleus. So the capsid itself is too big to get into the nuclear pore, but the DNA is not, and this is how we break up the capsid to get the DNA in. For poliovirus, it's simply the receptor that causes a pore to form between the virus and the cell. So here we have a virus particle binding a receptor. The receptor interaction causes a conformational change in the particle that releases the RNA very soon after the vesicles form near the plasma membrane. How is it released? Well, apparently a pore opens up on the capsid surface. Here's the 5-fold and the 3 and the 2-fold axis. That's a pore that opens up. It's catalyzed by the receptor, and it forms an umbilicus. So the membrane of the cell would be here. This is the virus particle, and the RNA would be coming out. So the receptor alone, interestingly, is able to do this. And noroviruses or caliciviruses do a similar thing. Here's the calicivirus particle. And when they engage their receptor, a portal is formed at one 5-fold axis of symmetry. So you can see a top-down view and a side view of that portal. Only one portal is formed. So your question before, how many receptors do you need? Well, you only need one 5-fold axis in both cases to be engaged to get uncoding. But how do you know which 5-fold axis to bind to, right? Because the RNA, there's only got 2 ends, and one end comes out first. So, you have 12 5-fold axes of symmetry. You have 2 of the ends are going to be in the right place. So how does the portal know where to form? I don't know. It's an interesting question. Maybe why the particle to P of U ratio is so bad, because the portal forms, no RNA here, move on, right? Maybe it forms in the wrong place, or maybe there's a better mechanism. It's an interesting question that needs to be solved. Reoviruses, I promised to tell you why that's cool, because they have the 2 shells, right? The outer shell and the inner shell. So what happens is these viruses attach to receptors, are taken up by endocytosis. And Most viruses get out of the endosome before it fuses with the lysosome because the lysosome has all kinds of enzymes in it, right? But the reoviruses stay in because those enzymes digest away the outer shell, and the resulting particle is hydrophobic, so it can poke right through the lysosome membrane, and in the cytoplasm it begins RNA synthesis. And plus that the enzymes in the lysosome remove these plugs, these orange plugs which plug up these turrets, and that's where the RNA is going to come out. So the outer shell is to protect the hydrophobicity of the particle, and when that's removed, it can get it out of the lysosome. So these are all the ways that I've told you that viruses can get into cells. We can get in at the cell membrane by fusion. We can get in by various kinds of endocytosis. We haven't talked about them, but basically different ways that vesicular transport occurs. And remember, the key is microtubule-mediated transport brings all these particles and vesicles into the cell. And for most DNA viruses and one RNA virus, influenza virus, they have to get in the nucleus. So the nucleus, of course, has pores that limit what can go in and out in terms of size. The ribonuclear protein of influenza virus is small enough to fit through the nucleopore. So as soon as those are released by fusion, they can go into the nucleus. But herpes virus and adenovirus are too big to get through the pore. So herpes virus sits on the pore with the portal right on the pore, and the DNA then shoots out. I told you how adenovirus gets broken up right on top of the pore. And then adeno-associated viruses, parvoviruses, those tiny ones, actually bind to the nuclear pore and cause a disruption in the membrane, and that's how they get into the nucleus. One last story I want to tell you about how receptors can control disease. So this is the story about rhinoviruses. There are 3 different kinds of rhinoviruses, A, B, and C. So A and B, they bind 2 different receptors, ICAM1 and low-density lipoprotein. And then the rhino C bind this protein called CDHR3. So here's ICAM1 bound to rhinovirus, and here's LDLR bound to rhinovirus. So the HRV-C viruses bind this molecule, CDHR3, an extended protein, to get into cells. Now, years ago, a single amino acid change in this protein, 529Y, was linked to increased surface production of the protein and increased risk of wheezing and hospitalization for childhood asthma. So they just noticed that kids who came in for asthma had this polymorphism in this protein, which led to higher levels. Then years later, it was discovered to be the rhinovirus C receptor. And in fact, cells that have 529Y have 10-fold higher levels of receptor, and they make more virus, and that's the risk factor for wheezing and asthma in kids. It's directly linked to virus infection. It's not, doesn't have anything to do with the receptor on its own. Okay, so 529Y is a risk factor for more severe rhino C. In 2013, there was an outbreak of rhino C in chimpanzees in Uganda. And that's one of them, Betty, who unfortunately died. They never see this virus, but it was brought to them by tourists. The tourists had the virus. They gave it to the chimps. All the chimps were 529Y. They sequenced their genome, which means they have high surface levels of the receptor and more severe disease. Only humans have 529C. Neanderthals do not. Denisovans do not. In other words, only humans have undergone selection for a severe illness that has selected for hosts that have 529C. And the Neanderthals and Denisovans either didn't encounter the virus or it wasn't long enough. But chimps never see it, so there's no selection in chimps for 529C. They all have 529Y, and if they encounter the virus via humans, they can get very sick. So a very interesting example of how the receptor can control the outcome of disease. All right. Now we're in a cell. We got in. We're going to start making RNA next time.